Welcome everyone. Welcome to the podcast of the third workshop on virus dynamics. We are here in Heidelberg, Germany. I'm here with Alan Perelson. I'm also here with Frederik, one of the organizers. Frederik, can you talk to us a bit about this workshop? What was the motivation for the workshop?
Yes, this workshop is actually the third one in a series that started in Frankfurt and was initiated by Udo Reichel and Catherine Beauchemin. And it basically aims at getting together theoreticians and also experimentalists working in the field of virus dynamics in order to enforce or enhance collaborations between these two disciplines.
Alan, you have been working in this field for a long time. Can you tell us the history of the field?
Yes, so my involvement in sort of the creation, I guess, of the name viral dynamics really starts in the mid-1990s, early to mid-1990s, where we started looking at HIV infection and trying to understand the response of HIV infection to drug therapy and what we could learn by studying the kinetics by which a patient would respond to such therapy. And in collaboration with David Ho, who's an MD, PhD, and head of something called the Aaron Diamond AIDS Research Center, he did very quantitative experiments during clinical trials of drugs. And I and some of my collaborating students and postdocs developed mathematical models that could be used to fit the clinical data, and it allowed us to come up with a set of mathematical equations that seemed to summarize what we observed in patients. And that initial work led to revealing a large number of different timescales of biological processes that underlie HIV infection. And over the years, we were able to establish there's at least 4 different timescales. So it's rather interesting, sort of as in physics, you perturb a system and you look at the response, and you can, depending upon how frequently you sample the data, you can identify different processes. And the process of modeling was to try and learn what the underlying biology is and then go back into the either clinical trials or experiments in the lab to validate all of that. And we were very successful at doing that. And that led to both more experiments by David Ho's group, but also other HIV researchers began to see the value of trying to use modeling to interpret observations within the context of treating HIV infection, but then also spread to other viral infections. So in 1998, I was approached by someone, a clinician who was studying hepatitis C virus, and we developed some fundamental models then for the dynamics of hepatitis C and how drug therapy would influence that. And then similar work began using hepatitis B virus. There was work in the UK on cytomegalovirus, and it's spread to a large number of other viruses. Influenza is one of the other ones that have been most studied, and it's been one of the hallmarks of this meeting, people who study and make mathematical models of influenza virus. Currently, we've expanded and are now working on viruses such as Zika virus and West Nile virus. Other groups are doing similar work with dengue virus. So, and in each of these cases, by doing mathematical modeling and looking at the actual dynamics, the time response of infected persons or animals to both the viral infection itself and to perturbations, one's been able to learn a lot about the basic biology. And by that, I mean, we were able to estimate how long cells that are infected by the virus live. How rapidly can the immune system of the host clear the virus? How rapidly does the virus replicate and infect new cells? As in epidemiology, we can calculate things called the basic reproductive number. So how many cells will one infected cell infect during its lifetime if there's no limitation in target cells? We were able to use that to try and design better experiments, better clinical trials for drugs. How long do you have to apply a drug to see a biological effect? Can we estimate, well, I should say we were able to estimate how efficiently some of these antiviral drugs were at being able to reduce the replication of the virus. It allowed us to both help design clinical trials and learn from them, and improve drug therapy in a very important way.
Can you tell us about your experience of working with clinicians and biologists?
Yes, I think it always needs a bit of time in order to find the same level of communication, so that everyone understands what are the possibilities but also challenges in either of the two disciplines, so that experimentalists basically know what kind of data mathematical modelers might need in order to get to some conclusive results, and the other way around, that mathematicians also at some point learn you cannot produce all the data you actually would like to have in order to build the best model that you are thinking of. So I think there it's a way where you truly have to work on trying to find an interdisciplinary environment where both people or both parties involved basically find the same level of communication and understand each other.
I agree with everything that Frederik has said. In the earlier days of this field, I mean, the difficulty was actually trying to find experimentalists or clinicians that would take modeling seriously, or that would even understand what was going on in modeling. So there were people who, at least in my experience, were generating very interesting data, but had no use for modelers. You call them up or go contact them, and basically they weren't interested in collaborating. But there were always sort of a few rare individuals, and they tended to be people with a slightly more quantitative background, possibly by their education, or who appreciated the complexities of their systems and realized by collecting data that they weren't getting the insights that they wanted and were willing to spend the time to at least talk with modelers. I think things have improved just given the successes we've had in this field. So now it's much easier, I think, for new investigators to make some of these collaborations.
So according to you, what are the key points to establish successful collaborations or to make the point that modeling can have an impact?
In my career, I've had the most successes when I first find something that's published from a group, having listened to talks, it sounds interesting, and they're already collecting quantitative data. I then try and come up, if it interests me, to try and do something on my own, making some preliminary models, maybe just digitizing data in a current paper, and then somehow then communicating with the experimental group some preliminary results, saying, we've taken some of your data, we've manipulated it a little bit, we think this is going on. It'd be interesting for us to both get the real data, but also, have you done X, Y, or Z, or are you willing? The model is suggesting we should do that, and sometimes that's more successful than just out of the blue, just writing someone and saying, I'd like to collaborate with you, or can you send me your data? They tend not to want to spend the time unless they see some benefit to themselves. I also always promise them co-authorship from the beginning. I said, if you provide us some data, if we ever do anything with it or write anything, we'll offer you and the people in your lab or collaborators who are instrumental in collecting the data co-authorship. We recognize, it's all of your funding and your livelihood depends on collecting this data, and it's a very valuable resource, and we respect that.
So do you think sometimes the work of experimental people are not so much valued by modelers, or vice versa?
No, I think, good modelers, who I would consider good modelers, are people who really look at the experimental data and try and make their models reflective of that. So I definitely value the experimentalists, and I think they're really crucial for making significant advances, at least in this type of field. I mean, and vice versa. I mean, experimentalists are a very heterogeneous group. Some see value in modeling and enhancing their work. Others don't see any value in that, and that depends on the area of experimentalization. And I think the previous success of trying to do quantitative work in that area.
Yes, I think also with regard to the development that the field of quantitative biology is taking at the moment, and also with the development of new experimental techniques, the requirement for mathematical modelers to really make an interpretation of the data that you actually obtain, and also to get a proper quantification of the data you obtain by these new experimental techniques is also seen by the experimentalists. So I think the field is really coming more together, that both parties know they depend on each other, and only then they can put the field forward.
How do you see the future of the field in terms of the skills which are going to be required for the new generation?
So especially for mathematical modelers, I would say that that more and more important becomes including and accounting for heterogeneity, stochasticity, and also all the type of information you acquire with single cell technologies and all this kind of techniques that really give you detailed information on a very low scale level, a minor scale level, and including that into your models, which of course will increase the complexity of your theoretical framework, is, I think, essential to still have the link between the experiment and the modeling, but also a challenge in some way, especially from the theoretical point of view.
I would agree with that, and I would add, in terms of the data that's becoming more and more important in this field, is also genomic data. I mean, sequencing has become much cheaper and easier, so we can actually see the variability, say, in the cases we're studying viruses, what the heterogeneity in the virus population is, and that can be exploited. One can also look at host response and try and correlate that with genomic studies to try and understand the variability in the hosts. And there's a whole field now of coupling virus dynamics with sequence data that's called phylodynamics, where one's looking at the phylogenies of the virus or the infecting pathogen, how it's evolving, and how the interactions with the immune system and the host are governing that.
All right, thank you for your insights. What do you enjoy in your research and teaching?
Of course, you always enjoy your research if you come to a point where you think, well, I have found a solution and I found now a model that works and fits with the data. That's something I really like about research. Of course, this is not usually always the case that you have these periods, but I think one important point is also that you have somehow some kind of freedom to look for the interesting questions and to really dig into a problem and try to solve it. I think there are not many areas of work where you can really focus on such things then.
Yeah, I have similar experiences. I get excited by solving problems, and I look at a lot of biological research as trying to find phenomena that are puzzling, and if I can get an insight into how some piece of biology works, and then do some theory and collaborations to either prove it correct or incorrect, sort of the whole process of seriously thinking about things is quite exciting, especially if you're looking at a problem in a way that no one, as far as you can tell, no one else has. And so you're sort of one or a few people in the world who are trying to understand X, and maybe you can do it, and maybe it leads to new things. But even if it doesn't, and you just all of a sudden understand something, I've always found that extraordinarily pleasing.
What motivates you to keep going when a model does not fit, or when a grant application is not successful, or when your hypothesis is actually not proven that model? What keeps you going?
Oh, the rare successes. And so, if you're doing good high-risk science, you're not going to be right all the time. You have to recognize that some of your ideas may not pan out. But as long as you continue to have a few good successes, I think that's enough to keep you going, or at least it is for me.
Yes, I think so. So for doing science, I think you have to be very consistent. And I think for doing science, you need to be able to deal with a high level of frustration when you come into problems where nothing works for long periods of time. But then, as Alan said, the successes that you have at some point, they can really keep you going. And if you bring this level of tolerance for frustration with you, then you can deal with this and go towards the successes.
So what would be a few words that you can say to motivate young researchers, PhD or new postdocs?
Well, I think you have to be excited by the science. I think you have to be excited by the science. You have to know enough about the biology or the mathematical techniques that making new discoveries interests you, excites you, drives you forward. The frustrations, of course, of finding funding. You may think you have a great idea and you write proposals and no one else thinks so, or at least not enough people to get you funded. maybe you persist a little bit and keep going, or maybe you just say, well, let's go find another problem. And I've had both experiences. It also helps to have a position that, as Frederik says, that gives you the freedom to do that. So if you have a university position and your salary is guaranteed based on your teaching, you usually can persevere and keep going. And if you lose funding, maybe you don't get summer salary 1 year, or maybe you don't get to hire a student that you want. But it still doesn't inhibit you from still trying to make discoveries, think about important problems. In my situation, it's a little worse because I don't have a teaching position and my salary is not guaranteed. So it puts me under more pressure to constantly keep up and think about good things and find good research problems.
You talk about mentoring and recruitment. How was it to recruit the good people or to mentor the good people?
I think it's really critical. If one wants both to pursue the research, especially if you have multiple ideas or like to do multiple things, you can't do it all by yourself. So it's important to get students or postdocs involved, not only just to do the research, but by forming a group, there are many, many more interactions. And I find it's much more valuable if you have an idea to discuss it with colleagues, even if they're students or postdocs. They tend to ask good questions. They say, I don't understand something, and it makes you think more deeply. Is there something wrong with your thinking or not? And just in the process of explaining what you're thinking about, I think, at least for me, it clarifies things. That's why I enjoy coming to meetings like this and giving talks and interacting with the audience and having them probe and ask questions. And sometimes they ask a question about something you've just not thought about, and it'll lead to new elaborations or directions. Also, just taking a young person and exposing them to interesting problems and seeing them get excited about it also excites me. So it reinforces. And that occurs, in some subfraction of the students and postdocs. Others just sort of come through and they do their project or something and they leave. So it isn't what they really want to do. Yeah. But it's the rare ones that get really excited, and then we can collaborate and do great things. Mm-hmm. Your current mentor, Rob DeBoer, was a postdoc of mine 28 years ago, I think, and we've collaborated ever since. It's been an enormous experience for both of us, and it continues. It's been marvelous.
Alan and Rob.
Yeah, I think so too. I can only agree. And in some way, teaching goes both ways, and then it's the most successful way of teaching. That not only you can inspire talented students to really follow their interests and work in certain fields, but also the other way around, like Alan said, that you learn new perspectives and also new ideas for certain problems, and thereby also get a better idea about the projects you're currently working on. So I think it's really crucial to find young people that have the interest and bring the interest to work in these areas of research.
Thank you very much, Alan and Frederik. This is the end of the podcast, and thank you very much for your time. Enjoy the rest of the conference.